again. I work at Thomson Mass Spectrometry Laboratory, and I will talk about mass spectrum, the pioneer and a new tool for petrochemical analysis. Oh, okay, thank you. The base of my research is mass spectrometry, and here we can see the website of the laboratory. I will explain a brief explanation about mass spectrometry. Mass spectrometry is like a big weighing scale that weighs a molecule. But to do this, the molecules need to be ionized. So they have to, we have a positive ion or negative ions. The base of basically the how does a mass spectrometry system work? During my presentation, I talk about mass spectrometry only MES. Okay. So first we introduce the sample. And this introduction can be by direct way or by chromatographic technique, like HPLC or GC. After this, we have the ionization source that we have different, it depends of the sample, and depends the substance that you have. And there are a lot of type of ionization source. Then we have the analyzer, and with the different way, it depends of the purpose. And then the detector. This part is in high vacuum, and the new technologies is growing up during these two parts, ionization source and analyzer. Mass spectrometry has some Nobel Prize, and it will begin in 1897 with J.J. Thomson, and in 1912 he created the first mass spectrometer. And from 1953 began new analyzer and new source, and until now, where we have ultra-high resolution analysis, that it's interest in petrochemistry. All of us know that oil of fossil origin is a complex mixture, and that has complex chemical composition. It took millions of years to be formed in sedimentary rocks, and became the main source of energy in modern world. We know also that crude oil has around 70% of hydrocarbon, around 30% of aromatic hydrocarbon, around 15% of resins and asphaltenes, and only 5% of polar compounds. That is the memory of the petroleum, of the oil. And is this part of this 5% that we have a group in our laboratory that work with apply this technique to analyze. So what is petroleomics? Is, in the broad sense, the study of all the components present on oil and how these components affect the properties and reactivity of a specific oil. So, with petroleomics by MES, we have the composition of the substance, and from them we can see their properties. Well, before show you the works, I can briefly tell about resolution and accuracy. What is resolution? Resolution is able to separate two ions that have approximated mass. And accuracy could be the evaluation from the error by this formula. And it's important, both resolution and accuracy, both are important for correct assignment of molecular formulas. So, higher the accuracy as nearest the experimental mass to theoretical mass. So, why it's important the resolution of the equipment? Because comparison between high and low resolution, with low resolution we see only a single peak, and we can't see the substance that are hidden inside this analysis. So with high resolution, with contaminants, for example, with high resolution, we can be showing two peaks. With low resolution, with contaminants, but only one peak. So it's very important that we have ultra high analysis with this technique. So, for example, here we have a spectrum. You can see that it's a ultra high resolution, 400,000 resolution. And in a window, you can see that between 0.2 Daltons, we can see all of these compounds it is with an excellent accuracy. Again, resolution accuracy, the importance of ultra-high resolution. We can see all this substance that we can see, and accuracy in the last decimal. Well, now talking about some works out of our laboratory. Here, it's a work that about biodegradation. And here, you use FT-ICR MS, ultra-high resolution technique in mass spectrometry. And we can see the profile of a non-biodegraded, and a mixture. And look at the zoom of the spectrum. We can see the oxygen, okay? We can see the biodegradation. Put in a class distribution. The abundance of O2 species is greater in biodegradation, in green, than non-biodegradation crude oils. The abundance of O2 species increase as the degree of biodegradation increase. And we can see it by these techniques. This work was a presentation ESI MS this year on American Society of Mass Spectrometry Conference in March, using this technique. And we can see with ultra-high resolution, we can see, besides having the accuracy mass, we can get the three analyses. We can see the number of carbons, the dBA, and the polar compounds, nitrogen, oxygen, and sulfur. So, analyze petroleomics by MS, analyze all of the oil. We can analyze about exploration, production, and refining. Okay, origin, maturation, all of the exploration, all of production, and all of refining. And you can use the term fingerprint. What is fingerprint? Fingerprint is like human fingerprint. We have MS fingerprint. So we obtain the spectrum. With this spectrum, we have the diagnostic ions. And with them, we can do the analytical characterization of the sample profile. So can you use direct insertion mass fingerprint to classify crude oil sample? Can you use this technique? Yes, we can do it. So, for example, we can, this work by our group, they analyze kerosene, gasoline, and diesel using fingerprint by polar marker. So, it's not this machine is not ultra high, but it's a low resolution. But with this product, we can see the difference between gasoline, kerosene, and diesel. Or, for example, if we have an additive, we can see the ion from this additive, the artificial marker. And for example, another example, profile of mixture of gas diesel in different proportion. So you can see the difference of the profile. And Também we can evaluation the oxidation, is the time? Okay. Evaluation of oxidation product in fatty acid. This part is exactly what I work, because we have some, we are in 55 persons in our group, and I work with biodiesel. So we can analyze with low technique evaluation of oxidation product in fatty acid extra. So for example, I can see is if the biodiesel is oxidized. So for example, in time of oxidation, above 10 hours, we can see dimers. In appearance, the secondary compounds. I can see the dimers and compounds from fission hydroperoxides. So I can see that the biodiesel is in inadequate conditions. So I use this technique in conclusion. You can see all the, applying this technique, we can see everything all about the oil of asphaltene, polar, structure, hydrocarbon, everything. And our group is some research of the petroleomic group of laboratories. On the left, we have the group that work with oil, and the right include me. We will work with biodiesel, applying this technique, and Dr. Marcos Eberlin is the coordinator of the laboratory. We are 55 researchers. Is this the view of our laboratory in Campinas, São Paulo, Brazil, and again, and our group. Thank you very much for your attention.
Thank you very much, Dr. Cunha. Any questions?
Okay, thank you. It's a very interesting talk. Did you only use the negative mode electrospray ionization as the ionization method? Yes. Because that will not ionize any saturated hydrocarbons.
Yes, but how can I say? With biodiesel, I can use, for example, because I analyze feedstock, so I can analyze in positive mode and negative mode. But with oil, normally negative mode, but I think that have worked that they use positive mode. I don't remember exactly what, but I know that they work. If you want after I can talk about it. I can give you the email of the people of the other group. But they use positive. I don't remember now exactly what. I can use with feedstock, but with oil, unfortunately, I don't know exactly what they work with positive on.
Okay, but so it's extremely challenging to ionize large saturated hydrocarbons, and there are only a few methods known today. So your talk really focused on just the 10% of the polar compounds.
Yes.
The 70% of the saturated hydrocarbons you cannot analyze using the methods you discussed.
Yes, yeah, I told this because nowadays they work with these polar compounds. The project, our Petrobras that we have, they work focus on polar compounds.
Sure, and that's really important. I just wanted to make sure that the audience knows that you couldn't analyze the 70% of the oil.
Seven. Okay, so like that. 5%.
Saturated hydrocarbons, which is 70%.
Okay, I put around, not exactly.
Okay, Dr. Jones.
Yeah, this question is stimulated by the previous question. I think the point being made was that only quite a small proportion of the material is so analysed. And I thought of an analogy with work on coal tars by MSGC, whatever, 15 to 20% of the coal tar is ever analysed. The rest just lodges in the capillary column, in the chromatography column. So it's an analogous difficulty. Yes, yes.
Any more questions? One more question, if anybody.
How expensive is this technique? How high throughput it can go?
I imagine that will be some question about this. If you have a high resolution, it's around 1,000,000 the equivalent. But with, for example, I analyze biodiesel, oil bidomes. Okay? That show a picture that they are presented in ASMS. It's very expensive, but all that
No, somebody said you can just MS using the any surface. You can do the MSDS, right?
yes, it's in the profile, for example.
Yeah, but you could become very cheap with the technique.
Of course, we have this proposal in the future.
Any more questions? No more? Okay, thank you very much.
Thank you very much.